All right, welcome to the last section of 7-3 notes. It's going to be on facilitated diffusion and active transport. In facilitated diffusion, and you have to remember that once again, in the process of diffusion, diffusion requires no energy. And it's said to help substances across the membrane. So when you did your coloring sheet, you colored proteins, and some of the proteins went all the way through the cell membrane from top to bottom. Some of the other ones were just kind of embedded in the top, and there are certain ones that go all the way through. These ones that go all the way through are called transport proteins. So transport proteins go all the way through the cell membrane. The cell membrane have protein channels that make it easy for certain molecules to cross the cell membrane. Because the particles can't move through the cell membrane, they can't move through the cell membrane on their own because they're maybe too big, so that's why they move through the protein channel. This movement, once again, does not require any energy. It is a form of passive transport, even though it's called facilitated diffusion. It uses transport proteins to move things from an area of high to an area of low concentration. Once again, it's moving from an area of high to an area of low concentration. Some examples of things that move through these particular protein channels are glucose or amino acids moving from the blood into a particular type of cell. So that would be an example of facilitated diffusion. Active transport, on the other hand, and the word active should ring a bell. Active requires energy. So in active transport, energy is needed to move particles. So active transport requires energy, or ATP, and is movement of materials in the opposite direction. So when we're talking about moving materials in the opposite direction, this means that the materials are going from a level of low concentration to a level of high concentration, or what we call against the concentration gradient. So the example that I used when we were talking about diffusion, I talked about putting a truck at the top of a hill, putting it in neutral, and it just kind of rolls down the hill. Well, in active transport, if the truck was sitting at the bottom of a hill and you needed to go up the top of the hill, you would need some sort of energy to push it from the bottom to the top. Same thing with going, if you're on a river and you're going against the current. If you're going with the current, your boat just floats down the river. If you're going against the current, you're going to need some sort of motorized or some oars in order to provide some energy to get that going from point A to point B. So once again, in active transport, they are moving things against the concentration gradient in the opposite direction. So once again, materials move against the concentration gradient. Active transport of small molecules is carried by transport proteins, or it's actually pumped from one side to the other. So the proteins are kind of changing shape in order to help these particles be pushed from one side to the other. And here would be an animation that shows you how these particular proteins pump molecules from one side to the other. So energy requiring pumps move calcium, potassium, and sodium across the cell membrane. Changes in protein shape play a role in the pumping process. which you can see from the animation. There are two forms of active transport. They are endocytosis and exocytosis. And from the prefixes, which I'm sure you love learning in English class, endo means into, and exo means out of. Cyto means cell. So endocytosis means into the cell. Exocytosis means out of the cell. So there's the fancy definition. Endocytosis is the process of taking material into the cell by means of infolding or pockets of the cell membrane. Once again, endo, cyto means into the cell. The pocket breaks loose from the outer portion of the cell membrane and forms a vacuole within the cytoplasm. And you know that I will have a picture of this on an upcoming slide. There are two types of endocytosis. The first type is called phagocytosis, and phago means cell eating. So this is the cell taking in food. So phagocytosis is the cell eating. And as you can see from the picture, there is a bacteria particle right here. It's being engulfed or being surrounded. It actually then forms as I said, a little vacuole, and then it breaks away and it goes into the cell where it ends up being digested. It's used to engulf large particles such as food, bacteria, etc. into what are called little vesicles. And these are the way that a lot of single-celled organisms are going to get their food, through phagocytosis. And here would be an example of a microscopic picture of a portion of the cell, and you can see that portion of the cell is extending itself, and this right here is the bacteria. And the cell is going to come out, this portion right here, and it's going to engulf the bacteria and then bring it back into the cell. Here would be another picture of phagocytosis happening. It's capturing a yeast cell, and the yeast cell is right here in yellow. This is the yeast. And this is the portion of the cell membrane that is going to extend around this yeast cell and then bring it back into the cell itself. The other one is pinocytosis. This means cell drinking. And of course, the way that I remember them is when you drink a lot, you have to pee. So you could say pinocytosis, but pinocytosis means cell drinking. And there is the same type of drawing where you can see all the fluid that's on the outside. And once again, it gets engulfed by part of the plasma or the cell membrane, forms a little vesicle, and then ends up going into the cell. And down here, this word cytosol, this means cytoplasm. It's another word for cytoplasm. Cytoplasm and cytosol mean the same thing. So here would be another animation of how this particular process of pinocytosis works. Tiny pockets form along the cell membrane. fill with liquid, and pinch off to form vacuoles. Or, once again, vacuoles are also called vesicles. So that's another word for vacuole, equals vesicle inside the cell. Exocytosis is the cell releasing large amounts of material from the cell. So this is where the cell would release things like waste. So this process is exactly the same, only it's going in the other direction. You can see where these transport vesicles have particles of waste inside of them. They move closer and closer to the cell membrane. They go up to the cell membrane and then they release. And in this case, it's actually particles of protein. They also can release waste in this fashion. The membrane of the vacuole surrounds the material, fuses with the cell membrane, forcing the contents out of the cell. So this particular animation shows how exocytosis works. There comes the vesicle full of the particles, moves up to the cell membrane, and then forces the contents out of the cell.